Section 17.5, we're going to be dealing with mutations. Sometimes mutations can occur within one nucleotide, or sometimes mutations can occur across several nucleotides. But nonetheless, they can, not always, but they can affect how a protein is assembled and consequently how a protein will function. And there's three types that we're going to be looking at to start with. The first would be a mutation. The second is point mutation, and the third is frame shift mutations. So to start with, questions 71 through 74 are quite basic, and it's more or less explaining what each of these terms is and a few examples of how frame shift could occur. So looking at 71, what is mutations in terms of molecular genetics? Simply it's this: it's changes in the genetic material of a cell or virus. So mutations are changes in the genetic material of a cell or virus. So that would be talking about changes in the DNA, changes in nucleotides. Mutations kind of includes a lot of different things. Point mutations, looking at 72. Point mutations are chemical changes in just one single base pair of a gene. So if you have a strand of DNA and a strand of DNA, so that double strand, and normally you would have A matching with T, A would be matching with G, which is not logical, doesn't make sense. So that would be one place on the DNA or one point, hence why it's called a point mutation. And the last one is a frame shift mutation. And a frame shift we had spoken about previously, and a frame shift occurs when the triplet code, the codons that we have on a strand of DNA, they're read inappropriately. They're not read correctly. And as a result, the rest of the DNA strand is read incorrectly. Kind of like if you're reading a book, you start at the first letter of every single word. Well, imagine what that book would change, like that sentence in each book would change, like if you, instead of we're reading from the first letter, you're reading from like the third letter in. So, aim, shit, ftm, yuda, tayo, nts. So it doesn't make a lot of sense. You do need to start at the first codon, read the remaining three codons that follow it, or three nucleotides making up the full codon for it to make any kind of sense. And moving into question 74, ways that frame shift mutations can occur. Two ways would be an insertion of a nucleotide or sequence of nucleotides, or deletion. So if we have a strand of DNA, add in an extra nucleotide, and that changes a codon and every codon thereafter. Or delete a nucleotide, which will change that particular codon and every nucleotide that occurs after it. So let's look at an example of a point mutation, which, by the way, there's no direct question in the outline that's tied to this. This is something you're going to want to definitely know. This is definitely testable material. So you might want to take note, even though there's no direct question in the reading outline tied to it. So let's look at a point mutation here. CGTATA, that's a sample strand of DNA, and we can see that we have a missense over here. We have GC, just like over here, but we have a flip-flop of this specific point on this secondary strand of DNA and TA right here. So these two are flipped around, TA and AT. Why is this a big deal? This still matches, right? Well, let's check out the resulting mRNA from each of these strands of DNA. Keep in mind that mRNA, it always will follow the one template strand of DNA. So let's check it out. GAA will be the result from this top template strand. Why is it the template strand? Why is 3 to 5 the template strand? because mRNA moves towards the 3' end as it builds. So this would be the template stranded CTT. So our complementary codon in mRNA would be GAA. Thinking to that codon chart we've been looking at with all the different amino acids, this codon would code for the amino acid glue. And this is used in hemoglobin. Over on this side, however, by switching those two around at that particular point, CAAT would be transcribed into GUA instead of GAA. This one little change would create a different amino acid, VAL. And in this case, if we're looking at hemoglobin, that one little difference will be the difference between a person having sickle cell anemia or normal hemoglobin. In sickle cell anemia, guys, it could be advantageous, although it's deadly. And if we think about why, it was typically found in African Americans and not so much in the European race. And sickle cell anemia is actually a prevention against malaria carried by mosquitoes, which is very, very high in Sub-Africa. So this might actually be why this genetic change occurred. It wasn't necessarily to cause a problem. Sorry to go on this tangent, but it actually could have been an evolutionary response to a scenario going on many years ago. To prevent that particular disease, malaria. So at any point, it's not good anymore. We don't need this particular evolutionary trait, and it will kill people very young. Sickle cell, the hemoglobin has an irregular shape to it, which can cause a lot of blood clotting. But at any point, this particular point mutation. Not much of a difference here, but a very big difference here, and the resulting protein, normal hemoglobin versus sickle cell. So what else can we maybe take note on over point mutations? The general categories, and there's two. There's base pair substitutions, and there's base pair insertions or deletions. The base pair substitution, that would be similar to what we saw going on with the sickle cell. Instead of having this TA combination, we substitute it for this AT combination. That would be one type of substitution, or remove this nucleotide sequence in general. Maybe instead of having TA, we have another CG. Substitute this combination with a different combination. And although these are the same nucleotides, they are in a different order, so they are now different. That is a substitution. And the second one would be inserting or deleting a base pair. And looking back at this, an insertion would be, okay, instead of CG, TA, at this point right here, let's put in another CG. So this codon would become CG, TA. This TA would be bumped this way. It would now be part of the next codon. That would be an insertion. A deletion would be, take your eraser, let's erase this TA right here, and now the next nucleotide would become part of this codon. So it would be CG, TA, and then whatever this next base pair combination would be. That would be the result of a deletion. So we're going to be putting a bunch of this stuff together now, looking at questions 74, 75, and 76. I'm going to be kind of skipping over this slide because we're going to be looking at each of these pictures larger scale. So we're going to start just by looking at this top strand really quick here, and then we'll move to this slide here. This top portion in blue is a sample strand of DNA. It's wild type, and wild type means that's normal DNA. This would be the normal mRNA produced from the wild type DNA, and this would be the normal polypeptide produced from the mRNA. So let's take a look when a silent mutation occurs within the DNA. Then we'll be looking at missense and nonsense. and then we'll be looking at different types of frame shifts. So our first example is going to be of a silent mutation which has no effect on amino acid sequence. Looking at our wild type polypeptide in DNA, we can see the last amino acid is GLY, which is coded by the mRNA codon of GGC, which is coded by the DNA sequence of CCG. Well, right here we have a substitution. We have A in place of the G, which was up here. So we swipe out GC and we substitute in AT. So we have CCA now instead of CCG. Well, what's the result? We have an mRNA codon of GGU instead of GGC. The result? Nothing. The polypeptide receives the exact same amino acid that it would before, GLY and GLY. Well, why is this? Because GGU and GGC are two codons that code for the exact same amino acid. Recall that some amino acids have several different codons that code for the exact same thing. So in this case, we get the same amino acid. This substitution is harmless. Next, we're going to be looking at a point mutation that results in a missense. And we can see the point mutation or substitution, T in place of the C up above. Looking at the same amino acid, the last one, and this point mutation, the substitution part would change the mRNA AGC from GGC, which would result in a different amino acid being popped on. This would result in what is called a missense mutation, because now we have a different polypeptide than we would before, and the function of this polypeptide, well, it could vary. Maybe this will be a different protein sequence, maybe it will be non-functional entirely. The results of it, they're not always the same. Will this function or will it not? Well, it depends. But this would be a missense. It's not what we were supposed to get. And the last type of point mutation involves a substitution at a different location. We're going to be looking at the second amino acid now, which was coded by TTC. And here we're going to substitute out A in place of the T. So now the DNA reads ATC, which results in the transcription of the codon UAG, which happens to be a stop codon. So we have our AUG, our start codon, followed right behind by a stop codon. So we have really nothing, which is nonsense. Hence the name a nonsense mutation. So we've talked about a couple different types of point mutations. Point mutations are the result of various substitutions. So keep that stuff in mind. Looking at our timer here, we're about 13.5 minutes in. I'm going to have to stop this video and pick up with the frame shifts on the next.